Seminar has got it. So actually this seminar has a quite long history. I was supposed to give this seminar, I think in January 2019. And then a father of one of the researchers passed away, and it was postponed to March. And in March, the Corona era started, so it was canceled. So I was very happy when Udi and Michael asked me to come and give a talk. So I have a chance to present this talk at last. So today I'm going to talk about remediation and hydrological implications of oil-induced soil hydrophobicity in the Evrona basin. And I would first like to acknowledge my co-authors. The first is Zen Glee. This is the master student who worked on the project, and most of the results that you will see, it's her work. Also involved are the Evrona and the Gilboa Aryeh from the Sde Boker campus, Ben Gurion University, Faina Gelman and On Kruvi from the Geological Survey of Israel, Garbi Gordon and Uri Shavit from the Technion, and last but not least, Ilan Stavi from the Dead Sea and Arava Science Center. So let's begin. Nothing happens. Why doesn't let me move, go? לא, משום מה. איפה כבר? I need help here. Okay, how did you do it? I see. So our story begins on the 3rd of December 2014. At this night, there was a breach in an oil pipeline, a Katsa pipeline, and about 5 million liters of crude oil leaked from this pipeline next to Be'er Ora Junction, next to Elat. I imagine that most of you know this. And the oil flew past the highway 90, and flew along the braided stream of the Evrona Nature Reserve, as you see here in this very nice figure. I don't have a cursor. Okay. So although the oil flew to about 5 kilometers in distance, its depth was quite shallow in most of the places. It was within a few millimeters to 30 centimeters. And right after this spill, the immediate concerns were that the oil will flow to the Gulf of Aqaba. And we all know that there are these magnificent coral reefs there. So oil in the coral reefs, not good. Also, there was a concern that it will flow to Jordan. And also, there was a concern that it will cause air contamination and maybe have a negative effect on the population that lives nearby. So there were some emergency containment efforts that took place, such as building dams, excavating pool to collect the oil. Some of the oil was pumped. Some of the polluted soil was excavated and transferred to designated treatment facilities. But other than that, nothing was done up to 2 years later, where a single session of tillage was conducted with the thought that maybe if we plow the soil and aerate the soil, it will enhance oil degradation. More recently, about 2 years ago, there was some biological treatment that was conducted in the reserve. I will not talk about it today. Today, the focus of the talk is the hydrophobicity of the soil. So once the oil is in contact with the soil becomes hydrophobic and water repellent. And this is what we investigated in this research. So going back to the spill event, after the immediate containment efforts were made, everybody stopped, and the Nature and Park Authority, which is responsible for the reserve, started to investigate what should be done in this situation. And almost incidentally, they found out that there was another spill event in 1975. And this spill event happened very near the 2014 spill event, about a few hundred meters away. It was hardly investigated. All they could find is this report. This picture is from the report. And no active treatment was applied. And even today, if you know where to look for this oil, you can go remove some of the sediments that were deposited from 1975, and you can still see the contaminated soil. It has a very distinct color, and you can trust me, it also smells. So once we found out that there was another spill event, the immediate thought was, okay, we had a spill event 40 years ago. Everything is okay. I mean, there are plants, there are trees, animals. We don't see anything. We don't see even the pollution because it is covered. And then the Nature and Park Authority starts to look a little bit further, and they started to measure the densities of acacia tree. Acacia tree is a key species in the reserve, and a lot of animals depend on it. And in particular, they measured the density of young trees. And they found that the density of young trees is much smaller in the contaminated areas with respect to the density in clean area. So it means we have a significant problem here, because if we can't grow acacia trees on this area, it means that the whole ecological system is at threat. So it means that something has to be done. So from here, I want to give some, no, I will not give the background, but I want to show some more results about how plants can germinate and grow on this soil. So here I think we have a very distinctive pictures that show attempts to grow trees on this soil. So we have control soil and polluted soil from the 1975 and 2014 spill events. And you can see the big difference where almost nothing grows on the polluted soils, whereas there is a nice growth on the control soils. And also, if you look for other measures, such as the length of the root, you can see that the length of the root in the clean soil is much larger than the length of the root in the contaminated soil. So this was published by Tran and al, and I want to add that one of the co-authors of this publication is Einav Mazlish-Getty from the Jing Bank here in Vulcani. And she is doing very nice work up to now about how the hydrophobicity of the soil and the pollution affect the plants. So we are talking about Evrona. Let me give you some brief background on this area. So it's quite a unique area. It is used to be a classic playa, meaning it's kind of a closed basin with no drainage, but due to the tectonic uplifting, now the area is drained to the Gulf of Aqaba. and to the coral reefs, basically. The geomorphology is of ephemeral braided streams. The soil vary between fine desert alluvium clay loams with salted gypsum crust, since it was a playa, so all the water evaporated and the salt left behind. And the alluvium thickens and the grain size decreases towards the downstream. The climate, as we all know, the climate of Eilat is hyper-arid, extremely hot. We have just about 30 millimeters of rain per year, very high potential evaporation of 2,600 millimeters per year. Average temperature is 25 degrees, and the maximum surface temperature can reach 50 degrees. So basically, you can cook an egg on the soil surface. The groundwater system is of a brackish aquifer. Some of it is phreatic, some of it is confined with some perched layer. So quite complex groundwater setting. And this is the reserve, a nature reserve. So why is it a nature reserve? So we have some gems in the reserve. We have the largest herd of Dorcas gazelles in Israel, and almost each time that I go to Evrona, I see them. The Northeast dome palms are also there. I think that we all know this when we go to Eilat on the highway. And as I mentioned earlier, the outlet from the Evrona basin is the coral reef of the Gulf of Aqaba. And the last thing that we want is to pollute these coral reefs with oil. So these are all things that we want to consider when we think of possible treatments in the oil. So, as I mentioned, the soil that was polluted by oil became hydrophobic. And I know that most of you know what soil hydrophobicity is. I don't have to talk a lot about it. I just want to show you how severe this hydrophobicity is. So here is a clean soil. And do you see this? Okay. And we place some water drops on top of it. The soil is completely wettable and the water just penetrates into the soil. This is the contaminated soil. You see the color, it also smells. And we place a drop of water and the water just stands on top of the soil, nothing happened. Believe me, the water drops can stay on the soil surface for hours. And you can see how hydrophobic the soil is. Look at this almost perfect sphere here that are placed on top of this layer of contaminated soil that is placed on this microscope slide. So just to remind you why we don't like hydrophobic soils. So they reduce infiltration, and that's why they increase the surface runoff and erosion. And they induce preferential flow, and this is a problem because preferential pathway can be a path for contaminant transport. The contaminant can flow very fast in this preferential pathway. We don't have any good tools to properly predict where the contamination will come to, what will the concentration be. So this is a problem. We also know that hydrophobicity alters the hydraulic properties of the soil. We don't exactly know how yet. And as we've seen, hydrophobicity impaired plant growth and germination. So there was quite a lot of work done on hydrophobic soils. But most of the studies dealt with naturally occurring fire-induced or wastewater-related water repellency. And probably some of the folks that sit here worked on the hydrophobicity that is related to wastewater. Relatively, there is much less knowledge about hydrophobicity that is induced by oil contamination. So this gives us a good opportunity to learn about this kind of oil-induced hydrophobicity. And in Evrona, we don't have just one spill, but we have two spill events with 40 years between them. So we can see how this hydrophobicity changed over time. So this brings us to the objective of the talk. We'll go there. So the main objectives is to study the persistence and hydrological effects of oil-induced soil hydrophobicity in the Evrona, and to investigate the relation between hydrocarbon content and the severity of hydrophobicity. There are a few specific objectives. First, we wanted to monitor, to in-situ monitor the hydrophobicity and the water content in polluted and clean soils. And then we want to assess the natural attenuation of oil and the decrease in soil hydrophobicity over time under different conditions by conducting incubation experiments. And the last objective that I will just show a few results from is to quantify the in situ population of oil degrading bacteria and see if we can isolate bacteria that produce biosurfactants. So the first thing that we wanted to look at, this part of the research was led by Ilan Stavi from the Datsi and Arava Science Center. It was conducted by Gabi Gordon, that was a bachelor student from the Technion, and this is their final project. And we wanted to look into the hydrophobicity and compare the hydrophobicity in the 1975 spill event and in the 2014 spill events. So we chose 3 plots in the 1975 spill event, 3 plots in the 2014 spill event, and we had the same plots, but nearby in a clean soil. And we measure hydrophobicity by 3 different methods: the water drop penetration time, the molarity of ethanol droplet, and the drop contact angle. I will talk about it immediately. And we also measure the penetration resistance, which is a measure of the mechanical strength of the soil, and the hydraulic conductivity near saturation, which will give us some idea about infiltration capacity. So how do we evaluate hydrophobicity? We use these 3 quite simple approaches. The first 2 of them can be applied also on the field, and this was very attractive for us. So the first method is the water drop penetration time method. We just place a few drops of water on the soil surface and measure how much time it takes for these water drops to penetrate into the soil. If these drops takes more time to penetrate, it means that the soil is more hydrophobic. The second technique is called the molarity of ethanol droplet, MED, or critical surface tension, CST. And it's quite similar, but instead of water drops, we use aqueous solutions of ethanol, and we search for the concentration of ethanol that will allow these drops of solution to penetrate into the soil in 5 seconds. Now, the surface tension of ethanol is much lower than the surface tension of water. So if we need more ethanol, it means that the soil is more hydrophobic. Now we can report, instead of the concentration of ethanol, we can report the surface tension of the solution. And when we report the surface tension of the solution, it is called the critical surface tension. And the last method is the drop contact angle. And in this method, we just measure the contact angle between the drop and the soil surface. If this angle is larger, it means that the soil is more hydrophobic. In general, if the angle is larger than 90, the soil is considered hydrophobic. So here are the results in terms of the hydrophobicity. We see the water drop penetration time, critical surface tension, and drop contact angle in 3 layers of the soil surface, 5 centimeter depths and 10 centimeter depths. Red symbols are the soils from the 1975 spill sites, and blue symbols are from the 2014 spill site. Field symbols are contaminated soils, and empty symbols are the control soils. So let's first look at the control soils, and they all indicate completely wettable soil. The water drop penetration time is classified as wettable. The critical surface tension is basically the same as the surface tension of water, which means that water drops immediately penetrate into the soil, and the contact angle is 0. So no big surprise here. Let's look at the contaminated soils, and we can see that the contaminated soils are quite hydrophobic. The water drop penetration time is classified as strongly non-wettable to severely non-wettable, and the critical surface tension is much lower than the surface tension of water, and the contact angle is higher than 90 and can even reach 140 degrees. And another thing that we see is that in most of the depths, the results are very similar from the 1975 spill site and 2014 spill site, which means that although there were 40 years between these spill sites, the soil in the 1975 spill site remained as hydrophobic as the soil from the 2014 spill sites, which means that time didn't do much in terms of hydrophobicity. Now you must wonder, why is this difference on the soil surface between the 1975 and the 2014 spill sites? And this is simply because when time passes, new sediments are deposited onto the contaminated soil. So when we measure the properties of the soil surface in the 1975 spill site, we actually measure the properties of these new deposited sediments. So we get something that is more wettable. What about the conductivity? Here I show hydraulic conductivity. So we can see that in the soil surface, the hydraulic conductivity of the contaminated soil is much lower than the one of the clean soil, which means that infiltration is reduced. This is true for all the depths in the 1975 spill site, but in the 2014 site, the picture is a little bit more complex. It is about the same hydrophobicity at 5 centimeter depths, and the hydraulic conductivity in the 10 centimeter depth is a little bit higher than the one of the clean soil. This is something that is quite surprising, but we could find such evidence in other places in the literature. And we hypothesize that it may be related to aggregation of the soil because of the oil, but we haven't verified it. In terms of the penetration resistance, which is a measure of the mechanical strength of the soil, we see that the penetration resistance of the contaminated soils is much higher with respect to the penetration resistance of the clean soils. And this has an implication, for example, on the ability of seeds to germinate into the soil. So when a seed has to penetrate such a rough soil, I don't know the word, it will be much harder for it to penetrate with respect to this soil, which has mechanical strength that is much lower. Now, the next thing that we wanted to do is see how tillage affects soil hydrophobicity. So as I mentioned before, 2 years after the spill event, there was a single tillage session that was supposed to increase the aeration and enhance the degradation of the oil. So we checked the hydrophobicity in terms of the water drop penetration time and this critical surface tension in controlled clean soils, in contaminated tilled soil, and in contaminated soils which were not tilled. And without going to the details of the results, we can see that the results of the non-tilled and tilled soils are very similar. So in terms of tillage, it didn't do much in terms of the hydrophobicity. The soil remained as hydrophobic as it was. Yeah.
It looks like contaminated field. It becomes more hydrophobic.
Yeah, in some of the points it became more hydrophobic. So we don't have a clear explanation for this. Compaction? Yeah, it could be, but the penetration resistance decreased and the hydraulic conductivity decreased. So I'm not sure that this is the explanation, but as you see,
about changing the fractions, the type of fractions.
so yeah, so it could be also this, but in the big picture, we think that the tillage didn't work because the limiting factor in these kinds of environment, hyperarid environments, is not the supply of oxygen, but it's the lack of water. So if you don't water the soil, it will not enhance oil degradation. So we see that the soil is very hydrophobic, the hydrophobicity is very persistent, and we wanted to look what hydrophobicity does for water infiltration. So for this, we set up 2 monitoring stations, one in the upstream section of the reserve and one in the downstream section. And in each of these monitoring stations, we chose 2 streams adjacent to one another, one polluted and one is cleaned. And in each of the streams, we opened the trench and installed sensors that measure water content and temperatures. And here are the sections of these trenches. This is the contaminated trench and the clean trench. We installed an array of 12 sensors in each of this trench, where the density of the sensors was higher next to the soil surface and lower when we go deeper. We had 4 sensors at a 7 centimeter depth, 3 sensors at 15 centimeter depth, 2 at 28 centimeters, 2 at 60 centimeters, and 1 in 1 meter and 20 centimeters. So we installed the sensors. We also sampled the soil to obtain the grain size distribution. We also sampled the soil to obtain more properties such as bulk density and city water content. And we also characterized the hydrophobicity measurement, the hydrophobicity of the section. So when we finish to characterize the section, we just close the trench and we obtain the data by a data logger that transmits the data to the servers of the Geological Survey of Israel. So we continuously monitor the properties in these trenches. So today I'm going to show data just from the downstream section. These are the hydrophobicity of this section, and this is very hydrophobic, also in terms of the molarity of ethanol and the water drop penetration time. Very high hydrophobicity up to a depth of about 30 centimeters. And it also correlates with the concentration of oil that were measured using total petroleum hydrocarbon. We almost have 10,000 ppm next to the soil surface, and it decreases with depth. We can also detect some of the oil at a depth of 40 centimeters, and in deeper layer, we cannot detect the oil. So this is what we have to work with. Now let's look how it affects infiltration. So this is the rain that we had in the over more than 2 years from 2018 to 2020. And these are the water content that we monitored in the clean section. So as you see, in big rain events, these usually induce also flood events, and we can see an increase in the water content in these sections. So I want to zoom into 2 of such an event and to look closer. So the first event happened on October 20, 2018. And we can first look at what is going on in the clean section. So we see the mean water content that was measured by the sensors at a certain depths versus time. And what we can see here is more or less textbook infiltration. So we see a very sharp increase in water content that is registered in the most shallow sensors at a 7 centimeter depth. And then we go to the 15 centimeter depth, 30 centimeter depth, and the signal is attenuated at 60 centimeter depth. And we see that the wet in front comes after it comes to the shallowest sensor. Now let's look at the contaminated section. Here we don't see the same order. With the highest water content is obtained at 15 centimeters depths, and then at 30 centimeters, and only then we see the water content of the 7 centimeter depths. We can also see that the water contents are much lower than the ones that are reported for the clean soil. So if we try to estimate what is the total infiltration in this rain event, we reach 43 millimeters in the clean section and only 10 millimeters in the contaminated section, which means that infiltration was reduced by a factor of 4 due to hydrophobicity. And this is, I think, very significant if we consider things like the water budget, the recharge to the groundwater, and the water that is available to the plant. We can also look at the variance between sensors that are installed in the same depths and compare the variance of the sensor in the clean section and the sensor in the contaminated section. And we can say that in most of the depths, the variance in the contaminated section is much higher relative to the variance in the clean sections. So this may hint us for preferential flow that happens in the contaminated section and is responsible for such large variance between sensors that are installed at the same depth. Let's look at another rain event. So this rain event is from March 13, 2020. If you remember, it was a huge rain event, a huge storm. It actually destroyed much of the shore of Eilat, did some damage to the coral reef, and it flooded the whole Evrona basin. So again, we can look at the clean section, and we can see an abrupt increase in the water content that is measured in the shallowest depths of 7 centimeters and 15 centimeters. And then we see the response of the sensors in the 30 centimeters, 60 centimeters. This is the magenta line. And in this case, we also see an increase at a depth of 1 meter and 20 centimeters, which is delayed, which is okay. This is textbook. Now let's look at the contaminated section. And here we see a mess. The highest water content was registered at 30 centimeter depths. Only then we see the water content of the sensor at 7 centimeters, and we don't see much of a response at 15 centimeter depth. So somehow all the sensors that were installed at a depth of 15 centimeters missed this increasing water content. And when we calculate the total infiltration in this section, again, we see 55 millimeters of rain in the clean section versus 13.7 millimeters in the contaminated section. So again, we have a factor of 4 as it comes to infiltration. The clean section got 4 times water with respect to the contaminated section. In terms of the variance between sources at the same depth, this is less distinctive than we showed earlier, but still in most of the depths, the variance of the sensors in the contaminated stream is much higher than in the clean stream, which again may imply for preferential flow. The other thing I want to look at is the temperature. So our sensors measure both water content and temperature, and we can see the daily average temperature at the different depths, from 7 centimeters to 120 centimeters. And we can see very well the seasonal variation, which is decayed once we go to deeper layer, which is completely what we expected. Now, I want you to try to look into the details and compare the red line, which is the contaminated stream, and the blue line, which is the clean stream. It's hard to distinguish, but you can see that almost at all times, the red curve is on top of the blue curve, which means that the contaminated soil is a little hotter than the clean soil, and the mean temperature is higher in the polluted section by 0.6 to 1 degree Celsius. This is not a lot, but this may affect, for example, the behavior of animals, such as lizards that like to go to hotter place. And maybe it can adversely affect these lizards because the hotter place are the contaminated ones. So it's something that should be considered. Now I come to the last section of the talk. What is the time? Time to finish. Okay, 5 more minutes. So in this part, we wanted to look how different treatments affect soil hydrophobicity and the composition of and the amount of the hydrocarbons. So we took soil batches from the 1975 and 2014 spill sites, and we added water to 20% and 50% saturation. And some of the treatments we also added surfactants, and some we added nutrients, and in some we added nutrients and surfactants. And we periodically sampled the soil for both hydrocarbon and hydrophobicity. And this experiment was done during 1.5 years. So let's see the results. Here I show the hydrophobicity of all the treatments in terms of the molarity of ethanol droplet. Let's look at the treatments of 1975. So the blue line here is a control. So we basically just put the soil as is, nothing happened, no surprise. When we add nutrients and water to 20% saturation, we get quite a sharp decrease in hydrophobicity. When we add only water, we still get a decrease in hydrophobicity, but it is more gradual. At the end, we get more or less the same hydrophobicity. In the 2014 soil, we have more treatments. We see that when we add both surfactants, nutrients, and water to 20% saturation, we get the highest reduction or the fastest reduction in the hydrophobicity. This reduction is a little bit more gradual when we add only nutrients, and this, the turquoise line, is when we add only surfactants. And again, when we add water to 20% saturation, it takes much more time, but at the end we get more or less the same hydrophobicity. And now an interesting treatment is when we add water, but we add more water. We add water to 50% saturation. And here we don't see the same reduction in hydrophobicity, which means the 20% saturation is more effective than 50%. As far as Evrona concerns, this is actually good news because Evrona is hyper-arid and we don't want to change the environment too much. So if we have to add less water, it's better. So it wouldn't enhance the germination of invaded species, for example. So we see that we don't need a lot of water. 20% saturation is enough. We also measure the hydrophobicity in terms of the water drop penetration time, and we get more or less the same results, so I'm not going to repeat it. And we also wanted to see what is the evolution of the hydrocarbon. So we measured at first this ratio of pristane. Pristane is this molecule, and C17, which is a molecule of straight 17 carbon. Now, when the bacteria wants to consume hydrocarbon, they prefer to consume straight molecules. So they will start to consume the C17, this ratio will increase. And then when the C17 is depleted, this ratio will decrease back. So we are looking for these peaks, which are a sign of biodegradation. And we see these peaks in the 1975 treatments, a big peak when we add water and nutrients, and a smaller peak when we add just water. In the 2014 treatment, we see the same order of peaks, which means that biodegradation happens first when we add both nutrients and surfactants, and then when we add only nutrients, and then with surfactants, and then when we add only water to 20%. And this is exactly the order of the decrease in hydrophobicity that we saw in the previous slides. What about the amounts of hydrocarbons? So we measured it using total petroleum hydrocarbons, TPH. And again, we see a similar picture. We see that at first the TPH decreases when we add both nutrients and surfactants, and then with nutrients, then with surfactants, and then with water alone. So if we add just water, it is enough, but it will take much more time. At the end, in all treatments, we obtain about 40% decrease in the total petroleum hydrocarbons. In the 1975 soil, we obtain in the treatments with nutrients even more oil degradation. We can degrade 80% of the oil, which is, I think, nice. And we think that the reason for the difference between the two sites is that the microbial population in the 1975 site is more adapted to the oil, so it can degrade the oil faster. So I think I'm going to skip this slide of the microbiological work and let Zev talk about it. And I will go to the summary and conclusions. So the contaminated soil from the 1975 event is as hydrophobic as the one from the 2014 event, although 40 years passed between the first and the second spill events. Tillage didn't decrease soil hydrophobicity and only marginally improved penetration resistance and hydraulic conductivity. So not much gain here. And hydrophobicity considerably decreased infiltration in natural rain and flat events. We saw it in the section that we monitored. Preliminary observations suggest preferential flow at contaminated streams, and the temperatures are higher in the contaminated stream, and this may affect animal behavior. In the incubation experiments, we saw that a significant and concomitant oil attenuation and decrease in the hydrophobicity that was observed during the 1.5 year incubation experiment, yet the hydrophobicity decreased, but as far as hydrophobicity goes, these soils are very hydrophobic also after they were incubated. So there is still something to be done in order to improve the hydrophobicity. The addition of nutrients and surfactants enhanced biodegradation and the decrease of hydrophobicity, but it didn't affect the end results. Water is limiting, but 20% saturation is more efficient than 50% saturation. And I will just give the results of the microbial assays. So they found that native bacterial population has a natural attenuation potential and can produce biosurfactants. Lily found bacteria that is able to degrade oil, and some of the strains she isolated were also able to secrete biosurfactants, and this has the potential to enhance oil degradation on the field. So with that, I want to thank you very much. I want to thank the funding from the Ministry of Environmental Protection to the Nature and Park Authority for giving us the permit to work in the nature reserve. For our colleagues, Yoni Laron and Ofer Dahan, who worked at the same site with us, and to all the guys from the Geological Survey of Israel who came with me to the field and helped and installed the sensor, worked in the trenches, and so on. So thank you very much, all of you, and I'll be happy to take some questions.
Thanks, thank you. For the Zoom speakers, if you have questions, please write them in the chat box and we will try to address them. And anybody here, right?
So we think there was some kind of factor that limited further degradation of the oil. We don't know what is this factor. It could be micronutrients or it was batches. So it may be some byproduct of the degradation that was poisonous for the bacterial population, but we don't know the reason yet.
One, in the beginning, you had shown effective measurement by water filtration time and EV and contact in depth. My impression that water filtration time and EV, they strongly correlate. When one is growing, second is decreasing, or maybe it was a critical angle. critical angle shows opposite different results.
Let's
Just
see.
really follow it.
I think you're talking about critical surface tension.
In my opinion, what we call
This one.
yeah. While water drop penetration time is increasing in depth. Contact angle from the right side, from the right side.
Right.
Yeah. It's contact angle.
Yeah.
So it means according to the DPT and the depth, it becomes more hydrophobic, but according to DCA, it becomes very hydrophobic. And then DCA is not changed and DPT is changed. There's no correlation between different measurements.
Yeah, so you're correct. We didn't find good correlation between the drop contact angle. And also, I didn't show, but we have results that were done in the lab in Lily's experiment. And we found that the drop contact angle is less sensitive in respect to the water drop penetration time and MED. We don't exactly know why. It may be related to the fact that this is done in the lab. It's done on, you put a very thin layer on a side of a microscope. So maybe it depends on the properties of this layer.
Second question is about using the ratio of pristine to C17. First of all, how did you measure that one in total contact? And secondly, why did you choose the pristine in C70? I understand that one is branched, the second is linear. But you have so many linear defense, and you have aromatics, you have phenol, naphthalene, that do not degrade. So what does it give us of the one?
So it gives us.
Can you repeat the questions, please?
So the question was, why did I choose the ratio between pristine and C17 and not other indicators? So as far as I know, it's not really my field, but more Faina's. This is a common marker for biodegradation. And when you start biodegradation, you first see the degradation of the lighter compounds. So this may signal the start of the biodegradation process.
How was measured total?
So the total petroleum was measured by, I think GCFOT. It wasn't GCIR.
So they just measure evaporation. How do you know the total content? Technically, you have to hit it probably.
So I'm not familiar with the technical details. I can get the answers from Faina, but sorry.
You have two questions in the chat box. Did you check the profile? Drova is asking, did you check the profile of the compounds? And did you actually end up with the undegraded polyaromatics?
So we checked the lighter compounds using GCMS, but not the heavier ones. And what was the other?
Did you actually end up with the undegraded polyaromatics?
So I don't think we ended up using only these heavy substances. We still see in the GCMS some lighter ones. It's not all degraded.
Okay, and another question by Guy. Why was 50% MC less effective than 20 MC in reducing hydrophobicity?
So our hypothesis is that bacteria has an optimal water content where they work best. And maybe 50% was too much, and maybe it induced some limitation in oxygen.
Is there any practical treatment that going to apply for this?
Okay. So Zeev want to continue and look into the bacteria that produce biosurfactants. because this can, if we cultivate them and then apply it on the field, it can enhance oil biodegradation. In terms, and one more thing is that we, in the future research, Gilboa and I want to look into the effect of introducing biosurfactants to the soil and look into the chemical interactions between such soils and the biosurfactants and how they can work together and reduce hydrophobicity.
Irrigation, this general irrigation was considered, is it possible?
So, okay, so I didn't talk about it, but in Evrona, the Nature and Park Authority, I think that Rotem knows maybe better than I. Some commercial treatment was applied. We don't know exactly the details because it's commercial, but it consists of irrigation and then introducing some kind of surfactants, and then they introduced some bacteria. I think there were a few cycles. Yeah, maybe, not sure. And first they did the pilot and it was able to degrade more than 70% of the oil. But when they applied it in the field scale, they got then less than 40% degradation. So they didn't get what they expected to get. And now they are considering changing the polluted soil with clean one. So it's complicated.
It's not a major issue of your talk, but at the beginning you showed this germination test with about, let's say, 98% reduction of germination in the 75 and 2014 polluted soils. And then you concluded that it's hydrophobicity and preferential flow based on what?
No, I didn't conclude that it's all about hydrophobicity. It's partly because of hydrophobicity, but it could also be because of some toxic effects of the oil.
I don't believe that hydrophobicity will kill all the plants.
Yeah, no, I don't think it's
Since
all.
you try to drink crude oil instead of water.
I don't, yeah, I agree. I don't think we can relate all the effect to hydrophobicity.
There is also one comment here, by Uri. One should differentiate between the behavior of hydrophobic soils and actual damage to the reserve, as the vast majority of the soil was not polluted and the waterways are very thermal, so that the pollution of a waterway means nothing regarding the water pathway in the future.
So I guess it's a matter of scales. So if we have a tree that now is the root system of this tree sits in the polluted stream, this tree can suffer. Maybe if you are talking about the water balance of the whole reserve, maybe this is less significant, but I think it's something that is worth checking.
Maybe you check the Did you check the stability and how it affects with these properties?
No, we haven't looked at it, but yeah, it can be very interesting.
A little bit technical question of it, translating the TDR readings to water contents in the polluted soil, you use a so-called special calibration curve for four-phase soil, according to the different configuration of the soil.
No, we haven't used it because
the same calibration curve?
Yeah, the same calibration.
So you are aware that for the same dielectric constant, the water content will be higher for hydrophilic rather than.
Yeah, I am aware that there could be some change, but when you look, if you consider this simple mixing model that concerns the fraction of each of the phases. We don't have really an oil phase there. The oil is soaked onto the soil grain.
No, this is minor, but just because it's hydrophobic, the more continuous phase is air. In the hydrophilic soil, the more continuous phase is water. If water is more continuous and the air is encapsulated, you get a much higher electric constant.
I see.
There's a guy named Alex Fuhrman in Technion.
No, I don't know him.
When you talk to him,
I actually talked to him.
he understood that Nimrod.
Yeah, but Nimrod worked on completely different.
No, but I'm just telling you, if the soil is hydrophilic, so water are attached to the solids and the air is more entrained, right? More encapsulated by the water. So if in the background is water with 80 and in the inclusion is air of 1, the effective dielectric is several times higher than if the background is air, like in hydrophobic soil, and the water are encapsulated. There's also articles by Friedman.
Who's free? Oh, just kidding. So yeah, I'm going to look at it just from a first impression when we looked at readings from TDR versus water content reading just from the oven. They were the same. There was no.
Try to measure also the runoff to just get a mass balance, a rough evaluation of the infiltration, but just using the runoff from the rainfall.
Yoni Laron measured it. I don't believe that we can close a mass balance because these things always do not close. But yeah, we can do some work with Yoni Laron.
Okay.
Okay.
Thanks.
Thank you, guys.
Thank you.